Okay, and Thomas Schlange will speak about the standardization of liquid biopsy at the European level. And the title is quite long, so I will let him explain the story.
Let's press the button. Okay, sounds much better. Okay, thank you very much to the organizers for inviting me over to give this presentation on our Cancer ID Consortium, which deals with liquid biopsy and its standardization. So forget about the long title, because it also implies that I have answers for you, and I would say we are rather on a track to find answers, and this is what I would like to show you. Myself, I'm the FPS, or the industry coordinator of this IMI consortium. I'm a biomarker scientist, which doesn't mean a whole lot. I'm a chemist by training, so I would rather consider myself a biomarker or biobench user or customer, rather than somebody who is leading a repository in that sense. Okay, so let's see. Liquid biopsy, for those who are probably not so familiar with it, in the cancer setting usually is referred to as circulating tumor cells, circulating free tumor DNA, and microRNAs, which are not cell-bound. You may also put extracellular vesicles or exosomes in this, which we didn't do, and this is probably also a nomenclature thing, because in the end we will also work with exosomes. So what is the benefit of having a liquid biopsy as compared to a standard biopsy? Obviously, it's minimally invasive. It can be done in a setting where there is no primary tumor or resectable metastasis present. It enables you to do a longitudinal monitoring, because it's a low burden to the patient. You can do that every month more or less and get information from the bloodstream that comes from the tumor. It is already being utilized in the clinic. I'll come back to that point a little bit later. There may be a possibility for functional testing as well when it comes to circulating tumor cells, and you isolate them. But this is not so much in the focus of what we are doing, actually. Okay. So for drug development, and in the end I'm working in industry, so that's what we are ultimately interested in. What can we do with it? So we are always looking for good biomarkers in a global sense that enable us to select the right patient population for a given drug. And it's quite obvious in the ages of personalized medicine that there is no one fits all in any given indication, and this is particularly true for cancer, of course. The other thing that is very important for drug development is to show early signs of efficacy and to demonstrate that you're really hitting on the target and on the pathway you're interested in. And that's another thing called predictive markers that is of high relevance. And of course, for doing therapy decisions, identification, early on identification of treatment resistance or developing drug resistance is very crucial. Not so interesting in this respect is the target validation or the identification of new targets, just to make that clear. Obviously, what we are talking about is very dilute. So when you look in the bloodstream and you're looking at circulating DNA, you may be familiar that this is being used for prenatal testing as well, but it's only a minor fraction, of course, of the DNA you will find in the bloodstream. So what you need is obviously high sensitivity. And this is also particularly true for the rare cells, the circulating tumor cells we're looking at, because usually in the blood you will find them even at advanced stages in an abundance of like 1 in 1 million of leukocytes. So you're really looking at something that is really, really rare. Of course, whenever you want to use something in multi-centered clinical studies, it needs to be accepted by the regulators. That was another motivation why we thought we should have a deeper dive into liquid biopsy and how to use it. And of course, when you want to use it in a multi-center clinical study, you need to have a way to standardize and harmonize the protocol, so everybody all over the world is basically doing the same thing, which is not trivial. I'll also come back to that point in a minute. So where are we currently in the clinic? For circulating tumor cells, there is a gold standard, if you want, which is the cell search system. It is based on the expression of a surface marker called AbCam, which is expressed by epithelial cells. And the model is, of course, that most of the tumor cells in the bloodstream will express AbCam, which is not entirely true for all the indications. And you may be aware that there's something called epithelium-mesenchymal transition. So this marker can be lost in the course of the disease, or it's not even present in some disease. So what you want to do when you find that you probably do not have enough circulating tumor cells in the standard blood draw, there are two ways. Either you look for additional markers to kind of get a bigger population of potentially present circulating tumor cells, and this is being done by some of the technologies. The other thing, obviously, is to increase the blood volume you screen, and we also have some of the technologies in our consortium which try to do that. Either using an in vivo device that is inserted in the vein in the arm and basically collect cells from the flow through, or diagnostic leukapheresis, which in theory allows you to screen the whole blood volume for circulating tumor cells. Ultimately, when in industry we sit together and thought about implementing liquid biopsy in our clinical studies, we thought, well, it would be nice to have at least 50 living circulating tumor cells from at least 50% of the patients. And just up front, without going into further detail, that's not where we are. There will be some or much more further development needed. And this is also lengthy procedures like the DLA, for example, to screen large blood volumes, and whether this will be feasible at some point beyond a stage of research is not entirely clear yet. Okay, what about ctDNA? Currently, this is seen as much more feasible because it's much easier to work with it. And here we are back to the pre-analytical sample handling. You can take the plasma, treat it according to the standards, and freeze it down. This cannot be done when you look at circulating tumor cells, because they will only, even when you use a fixative, they will only be good for something like 4 days at the most. They can be biobanked to a certain degree, which we are also exploring together with IBBL in Luxembourg, for example. But of course, for a standard test, this is not what you want to do, because we're also looking at short turnaround times. For ctDNA, as I said, it's easier, and the year before last year, a test has been granted approval by the FDA, looking for EGF receptor mutations that render a patient insensitive to first-generation EGF receptor tyrosine kinase inhibitors. So this can really guide treatment decisions. You can really choose another compound based on the result of this liquid biopsy result, and that's what we want to do. So this is really the kind of predictive assay we are looking at. Obviously, a single mutation in a single gene may not be enough. So what you want to do, for example, NGS. And of course, people are doing that. But just to let you know why we feel we need to do a lot more in terms of standardization and to understand much more what a single assay is capable of showing us. This is an example from a recent publication in JAMA Oncology, where a clinical lab sent out a blood sample from a patient or different patients to two providers of NGS in liquid biopsy, and that was the result. So without going into much detail here, you can see each column is a separate patient. The red dots you see are discordant results that were found with one assay that was also covered by the other assay. The blue ones are mutations that have been found that were not covered by the other assay, and only the green dots there are actually mutations that were covered and found by both assays. And that is a problem, as you can see, because there is no ground truth, obviously. So what is the better assay? What is the therapeutically meaningful information we're getting from this assay? So we thought at industry, when we started the whole discussion on having a joint project on this, goes back to 2013 actually, what can we do? And the Innovative Medicines Initiative, IMI, actually provides a platform with standardized legal procedures, etc., and a framework contract scheme that allows addressing such pre-competitive questions together with academic and clinical labs. So just for those of you who are probably not familiar with IMI, it's a public-private partnership. 50% of the money comes from the EU Commission, 50% comes from the FPI industry in Europe. It's pretty big. So we are still part of IMI 1, which had a budget of 2 billion euros in total for a 5-year period. We are now into the IMI 2 period, which will last until 2024, and that's even bigger. And the nice thing when it comes to diagnostics is it's not only FPR companies now involved in this, but also diagnostic companies. Okay, so we set up a call text basically, and the winning consortium, after a lengthy procedure how to select such consortia, was Cancer ID, together with the academic leads Klaus Pantel and Leonta Stappen. who are KOLs in the field and have a long track record of working on circulating tumor cells and also circulating tumor DNA and mRNA. Was established in 2015. It's a 5-year project, so it will last until the end of 2019. As you can see from the map, although it's kind of small, there's a lot of red dots, most of them in Europe, but we also have some partners in the US. Currently, we are 38 partners from all over the world. We have a budget that is relatively small for such a big effort, but nevertheless, that's what we got. Roughly 18 million euros, and Bayer is a major contributor to that, so that's why I'm standing in front of you presenting the project. Good, so key deliverables of this project include the establishment of criteria for identifying what is a CTC in the first place, because as I said, it may not only be the epithelial markers expressed, there may be EMT markers or others, and also to separate them from other EpCAM negative cells. So neutrophils, for example, tend to be CD45 negative, which is normally used as an exclusion marker for leukocytes, or endothelial cells, which can be big. So size exclusion methods may also pick up endothelial cells. So you need to identify really carefully what could be a CTC, and then in the end only looking for molecular alterations can really give you an answer or confirmation that you have a circulating tumor cells in hand. The same is true for the ctDNA, obviously, so we are trying to establish criteria for good practice here, and also for miRNA. We're looking into standards, we are looking into establishment of SOPs and harmonizing the protocols between the different sites. We're setting up a database which includes the clinical data of the patients, so we are trying to find concordance between the different marker classes and also correlations to the clinical progress of the disease in those patients, which can only be done in an exploratory way because we cannot really deal with setting up our own clinical studies. Ultimately, we would like to support the regulatory approval of liquid biopsy technologies, and that's why we reach out to regulators like the FDA and EMA as well and get their input. Probably of note as well, we are focusing, because we cannot do everything, on 2 indications, which is non-small cell lung cancer and metastatic breast cancer. So what we did, we are already like halfway through the project. So we started during the first 2 years setting up the different technologies, at least in 3 labs, so we can really compare between different sites, which I think is really, really crucial. And we have working groups for each and every biomarker class here. We have a work package for data management. I will not go into much detail there. And work package 4 is my major playground because somebody has to do the project management as well, of course. So in the technical evaluation phase, we started to look into spike-in artificial samples. That is true for ctDNA, but also for circulating tumor cells, and try to establish protocols that can also work with clinical samples or test those protocols if they are already available from the technology providers. This is true for lung cancer and breast cancer, obviously. So ideally, we are looking at technologies that can deal with all the indications you're interested in. And finally, and we are moving into this phase now, we are trying to look at clinical cohorts to implement liquid biopsy tests there. And currently, our focus is shifting a little bit towards the immuno-oncology drugs and whether liquid biopsy can really contribute to selecting the right patient who has a chance to respond to those treatments. We found, and that's why I have these little blue fields there, there is some overpromising when you look at the technologies that are sometimes advertised. And we found that it's very, very important that we keep up those working groups for the different biomarker classes and that we continue to further develop those protocols. It's not trivial. This starts from pre-analytical sample handling. This starts with storage times. This starts with different blood collection tubes, fixative, yes, no, shipment times, shipment temperature, mechanical stress introduced into the sample. So there's a lot of variables that can affect the quality of what you're finally getting. And this is certainly something we need to look into over the whole period of the program. So this again highlights what was the status when we started the project in liquid biopsy. So you have different input samples, plasma, serum, circulating tumor cells in different matrices, etc. You have different investigators at different sites. They have their protocol. They will tell you, you know, this works for us. We will not even try anything else. And well, that's the truth. So you have to convince them of even considering looking into something else. You have various protocols resulting from that. You have different output formats. and then you get a result. And this will be very difficult to compare to what anybody else did. So this is what we are trying to do. On the right-hand side, we are trying to bring this together. So we are defining the input sample, we are defining the protocol, we go to different sites, as I said, at least 3 sites for each and every technology. We have a consensus protocol and try to look at downstream analysis like bioinformatics as well to see where there are factors that influence the information that may in the end guide the therapy decision for the patient. As I said, we are talking to the regulators. We had a critical path innovation meeting with the FDA in 2015 already. And they basically gave us their impression on where the liquid biopsy field is and what they consider really crucial to further advance that. They found that a very concise definition of the context of use is really crucial for each and every assay, and still not every technology is there, I have to say. They asked us to show equivalence with existing technologies. So in the CTC field, for example, we should look into comparable data to the cell search system so that we really make sure that we pick up the relevant populations of circulating tumor cells. And they also suggested to look into the establishment of standards. And that's what we tried to do, so a little bit more quickly. One example is we came up with like a standard cell line panel where we have data on the cell size, AbCam expression, mutational data. These cells are freshly provided by ATCC, and they will be provided to all the participants basically so we can make sure that they all work with the same or at least a controlled passage number, because we know that passage number really introduces a lot of change into the cells. Of course, having different levels of AbCam expression and different cell sizes also enables us to compare technologies that look at AbCam positive cells, AbCam negative cells, and at, of course, a wider population of CTCs that may be present in the bloodstream. For the CTC ring studies that we initiated, we looked at the yield, of course, in the different, using the different technologies at the different sites, compared that to cell search, heterogeneity, I mentioned that, purity, so what is the background of white blood cells we're getting, which downstream analysis is possible based on the quality of the circulating tumor cells and the isolated DNA we're getting from that, and how reproducible the whole thing is. And this is one of the typical examples we're getting. This is comparing to blood collection tubes, the cell save and the transfix, and the recovery with cell search. On the left-hand side, you see an AbCam positive cell, and naturally this is being picked up by an AbCam selective technology like cell search. And on the right-hand side of the left graph, you see an AbCam negative cell line, and obviously this is not being picked up. That's nice. You see there is a certain degree of variation between the different sites. So 5 sites have done this experiment, but it's still kind of controllable. And the funny thing, or the interesting thing actually, and this is true for most of the technologies, you always end up around 50 to 60% of recovery. There seems to be like a law of nature in this. I don't know why this is, but that seems to be the yield you're getting. And on the right-hand side, you see a filtration technology provided by Siemens, and again comparing AbCam positive and negative cells, and what you find is what you expect. A filtration technology obviously picks up both, and you get benchmarking data for each and every technology, and we are doing that for microfluidics, etc., etc. Another important thing is to get rid of observer bias. So for the cell search system, it's 2 pathologists looking at the pictures being taken from the cells and analyzing or giving their votum whether it's a CTC or not. If they disagree, there will be a 3rd one. Of course, that's a tedious procedure. There is some observer bias you have with that, and that's why we now have an algorithm that has been developed by Leon Terstappen in Twente and his colleagues that at least gives guidance for what an automated system does. And the ultimate goal, obviously, is to do that fully automated. ctDNA, same thing. There are some guidelines already, and we have people in the audience like Uwe Oehlmüller from QIAGEN who influenced these ISO guidelines quite substantially already. And we are looking into different parameters that are given there, like for example storage at room temperature of the blood collection tubes, etc. And there are 2 data sets that show how important this is. So on the left-hand side, you can see a ring study that has been performed in the SPEEDIA 4P Horizon 2020 project. And this is genomic DNA that has been isolated from whole blood. And in panel A, you can see that this is basically the input material that has been done at a single site according to a standard protocol. This has then been distributed to different other sites, and that is what they send back after their extraction. So you can see there is a quite substantial degree of variation, and I would expect for circulating tumor DNA you would find similar pictures. And on the right-hand side, you can see from a publication what happens when you use different temperatures. And the take-home message from this is if you cool the sample in a Streck tube, for example, or if you heat it up, you will lose quality. You will either have hemolysis, so you will have larger fragments, or at least you get a higher variability in the fragments you're getting. And this, of course, is detrimental to the quality of the samples. So ring studies is a core element of what we're doing in Cancer ID. So for ctDNA, we look into different tubes and do a ring study on that with fixative, without fixative, whether double-spun plasma is superior to single-spun plasma. Why is that? We could just collect double-spun all the time because everybody's doing it. But if you think about the retrospective sample collections that are out there, you may be interested to go into that as well. And if you don't have a quantitative idea how much this affects the quality, you cannot work with it really. Okay, then the suitability of different technologies that look into mutations like NGS panels, for example, or digital droplet PCR. The sites are blinded, so they don't really know what they are getting. I think this is also important for proficiency testing here. And of course the bioinformatics. So if different people analyze the same data, will the result be the same? We haven't done it yet, but my prediction would be no. Okay, so I probably skipped that. This is the standards we are working with. So there are commercial standards based on shearing. We are also looking into mononucleosomal DNA, which is probably a little bit closer to what you find in a tumor patient. And we're doing the same thing for miRNA. So the concept is always the same. You go from pre-analytic down to the bioinformatic analysis and compare at every step what kind of results you're getting between the different sites. And this, just to finish that up, gives you some typical examples. So here we compared a QIAGEN kit on exosome isolation, the ExoRNA Easy kit, with ultracentrifugation, which is, of course, a standard technology for exosome analysis as well. And we found that the ExoRNA Easy kit is a bit superior here. But there are some miRNAs as well where you get a mixed picture, so you may even have to look at that because it may have to do with the exosome localization of the miRNA in the end. And this is comparing different extraction protocols or technologies as well. So from Exicon, QIAGEN, Norgen, etc. This is very colorful, so I like to show that. But this is just giving us benchmarking data for each and every site that participates in these studies, so that they know where they are compared to the others. And usually what you would expect is for each and every technology or protocol, you find a cloud where each site clusters. If this is not the case, there's something wrong, right? Okay, so some important points for consideration. I would like to use that as a summary and close with that. So I think for liquid biopsy, which is a highly potent concept and certainly something that has the potential to advance drug development and diagnostics in cancer patients substantially, we need to show that it can be really applied in the clinic. To that end, we need to standardize the pre-analytical sample handling. This needs to be crystal clear. The hands-on time in the samples needs to be minimized, particularly for the circulating tumor cells, so this is really crucial. We have to use standards to be able to benchmark the different technologies and to show that each and every lab participating in such analysis is really up to the standard where it should be. And there should be feasible downstream analysis technologies, because if you think about workflows that involve a lot of different technologies, this is costly, this is time consuming, and this cannot be accepted because the turnaround time needs to be short. I think this is really important. I didn't touch on that too much, but the cost, of course, is also an important aspect, because if you think for example, non-small cell lung cancer, the number of circulating tumor cells is really low. So what we do is we have this DLA procedure to have more cells. You need to enrich the cells, and then you need to basically get the single cell to get the information you're looking for. Is this a feasible workflow for a routine lab in the clinic, or is this a mere research kind of essay? I think from the cost, right now, the answer is quite clear. But we are really looking for assays that can be used in the clinic. So this needs to be the future development. And with that, I would like to thank you for your attention.
Thank you very much. I have one question regarding EBCAM. If you have a patient with metastasis, do you have changes in EBCAM expression between the primary tumor circulating cells and the one from the metastasis?
Yeah, you find you can find the whole variety basically of phenotypes. So you have patients where you don't see circulating tumor cells. You find them with EBCAM positive and EBCAM negative, and the same is true for the primary tumor. So it's heterogeneous. And also for the metastasis, it's clearly heterogeneous. So the metastasis can be quite far away, particularly after treatment, from the primary tumor. So you have to expect heterogeneity for sure.
Other questions? No, you have a microphone.
Could you please to the microphone.
I wanted to ask you about this last statement that was on your slide, the lower limit of detection and lower limit of quantitation for CDCs and ctDNA determines the size of patient population that can be eligible for liquid biopsy. So is there a lower limit, or what number do you need to work and establish these parameters?
That's a very good question, actually, because when you go to big meetings like the AACR, for example, you'll find a lot of fantastic posters where people say we find enough DNA in like 95–98% of the patients to do a ctDNA, next generation sequencing, or whatever assay they want to use. When I talk to clinicians who are quite experienced in that field as well, they tell me it's probably 60% or probably even lower. And when you go from like late stages, metastatic stages, down to stage 3 lung cancer, for example, stage 2 lung cancer, this drops down to 40%. Because usually these assays require an input of something between 10 to 25 nanograms per ml. And this is not what you really find in all indications and at all stages. So, and then it becomes like a numbers game, of course. So if you have like a low allelic frequency mutation, you need a certain input to have a chance to find it, right? So I think we need to be careful, particularly when it comes to things like early detection, to really promise people we can use ctDNA for such purposes. This may be over-promising to a certain degree. Higher sensitivity may help. We may find more if we look more carefully, but this is not where we are right now, in my opinion. Thank you.